pleasure today to introduce Ed Theriot, checking in from the University of Texas at Austin. Ed is he's the professor and director of the Department of Integrative Biology in the College of Natural Sciences there, as well as the Harold and Mary Bold professor of, and this is my favorite part, cryptogamic botany, words that we don't see as often as we used to. Ed's been at Texas for a good spell now, where he's continuing to make us all know that Stephanodiscus is the coolest genus. He's on board with that. Ed's gonna be talking to us today about species concepts and diatoms, and providing some context for how we view these different types of things critically. So take it away, Ed. Floor is all yours.
Okay. Can people see my see the PowerPoint?
Yep, we can.
Okay. So I took this, well, first of all, thank you for asking me to do this. I kept seeing the announcements for the webinar, and I've only recently, since I got asked to do this, only recently have I started following them. It does sound like really interesting topics, and I need to show up to more of them. Today we will be talking about, well, to be blunt, why I think cryptic species and worrying about them is a waste of time. But to be less antagonistic, I'm 69 years old, so I've got old person syndrome. I don't like anything anymore. But I thought I would approach this from the standpoint of, well, what is a species? I took this quote from an early paper by Gareth Nelson, a famous cladist, on species. In his view, species are just taxa. We'll review that more in a little bit. Okay, and now, earlier I could switch screens, but now I can't. There we go. So this is a more or less confrontational title, though not much. I do firmly believe this. I mean, I see so many papers. We all see papers about cryptic species, cryptic species, cryptic species. I think this is a distraction. There's many more interesting and important phenomena to understand about diatom species and speciation and evolution of diatoms. So this presentation is going to have three parts that addresses that and explains why I think so, why I think these things. First and foremost, something called a morphological species or a morphospecies, it doesn't exist in the sense, from a scientific perspective, it's not a testable hypothesis because it's an extremely vague statement about what a species is. As we'll discuss, basically a morphological species concept is authoritarian in 19th century. The words are there, sure, but it has nothing to do with actual science. Then I'm going to present a very simplified, and for any of you who are deeply into the what is a species literature, I hope it's acceptably simplified for you all. Again, this presentation is not about really what is a species, but I think I'm going to be able to present core ideas. And many of you are ecologists. I'm not being critical. Because there's a ton of literature on ecology that I don't know of. There's a ton of literature on what are species that I'm not even aware of, although I try to read a lot of it. So I'm going to give a simple presentation on three major concepts: biological species, phenetic species, and phylogenetic species. Almost cartoon versions, but you're going to see the real point here is that none of these what we call modern species concepts have anything to do with the type of data used. Excuse me. And then I'm going to review a couple of studies in Cyclostephanos and Stephanodiscus that have been done in my lab. We could perhaps pick a couple of others that tell the same story, but I'm focusing on these two because I think, not just because they're from my lab, but I think they're the most complete in addressing a number of fundamental issues about species. So parts 1 and 2, I apologize, are going to be a lot of words. The pretty diatom pictures are going to start with part 3. There's 2 words that I'm going to use that they're very much jargon. So I want to make sure we understand what I'm talking about. One is the word operational or operations or operationally. If I use that word, it's shorthand for saying, referring to the actual practice of some procedure. In this case, it's the operations of recognizing new species. Another word I'm going to use is canonical. The operations can be canonical or non-canonical. What canonical means in this situation is having a set of explicit assumptions, rules, or algorithms. In this case, tools like cladistics, cluster analysis, and or multivariate analysis, whether applied to morphological data or molecular data. The antonym non-canonical in this particular case, of course, it's what it sounds like. It doesn't have a set of rules, but I want to be clear that when we talk about non-canonical approaches to identifying a species, we're talking about vague approaches. You may have a particular approach, but Bob and Jane have different approaches. So it's vague, it's often authoritarian because I'm an expert in this group, I say so. Or that there's some unspecified combination of operations that are going to vary from instance to instance within a researcher's practice. So that's what I mean by non-canonical. We really don't understand. Yeah, I see these discontinuities, but we don't know where that vision came from. Before we can define a cryptic species, we need to know what a non-cryptic species is. So here we go with that discussion. I want to emphasize it's not species or not just the data type. What we have in this, one of the reasons that cryptic came about is we have what I'll call moleculose species. This is a picture of green algae higher classification, but you could write in species names here. It would be the same thing. But I picked this in part because Rick and I think it was Louise Lewis were doing this working classification of green plants, and they did a molecular analysis. Well, they came up with monophyletic streptophytes, monophyletic chlorophytes, and so forth and so on, and compared that to the morphological classification. But their comparison, like so many others, were comparing a concept. Yeah, it was molecular, but what other thing that was different about it is that there was formal analysis, formal phylogenetic analysis of these data, whereas the morpho classifications were based at that time on, still based largely on traditional approaches, an eclectic mix of non-canonical things. These look alike, they're in the same environment, and so forth and so on. So in a lot of ways, they were comparing apples to oranges, and not just in the data type, but in the philosophies of classification. And this is really important to remember. So let's get back to species. Ed, what's wrong with the morphological species concept? I use it, or I see it referred to a lot. Well, here's some statements. This is from a Diatom paper, and I'm purposefully not citing where some of these statements come from. One, because in fact, they're quite general. Two, I don't mean to be particularly critical of anyone. I am critical of the ideas, but I'm not meaning to be critical of anyone. So that's from a Diatom paper. I mentioned it's from a Diatom paper to show the relevance to diatomology. Morphospecies, the outcome of applying the morphological species concept, Sinsu Meyer, 1942. I read a lot of Meyer's work, most of it in his books. Here was a paper he published in 1999. It was easy to get so it was easy to pull it up on my screen. and read it. And he really hasn't changed it. He says the morphological species concept is equal to the, introduces another name here, typological species concept, in which degree of morphological difference was the species criterion, morphological species concept based on typological essentialism, and so forth and so on. And there's still, so morphospecies, the outcome of applying a very vague concept, Sinsu-Meyer 1942, is how you can read this. For example, typological, if you read the Diatom literature, sometimes it just means being close to the type specimen. And remember, he says here, degree of morphological difference. So a late good friend of mine, Hannelore Hawkinson, was a typologist. She viewed species as how much did it look like the type? Did it look enough like the type to be the same species, or did it look different from the type? Well, that's a personal judgment, frankly. Another meaning of typological, and I think this is what Meyer actually meant, was having some essential, undefined, essential characteristic or multiple characteristics of the group. And of course, both of these are in the eye of the beholder. No real definitions of those things. Morphospecies then are like body pictures. Famous statement by a US Supreme Court Justice when the issue of pornography and free speech was being discussed here in the US. He didn't know he couldn't define pornography, but he knew it when he saw it. A counter to that by another lawyer was, I know it when I see it, and someone else will know it when they see it, but what they see and what they know may or may not be what I see and what I know, and that's okay. This is, in a nutshell, morphospecies. It remains authoritarian, it remains a vague practice. So we're comparing, when we compare molecular species to morphological species, to morphospecies and the morphospecies concept, we're comparing not just the data, but we're comparing one grouping that's based on a formal system of analysis. We don't always agree on it, but if with the same data, we can all, and the same algorithm, we can all come to the same result. And that's not true for morphospecies. Back in 1926, a person named Reagan, C.T. Reagan, made this clear. In the opinion of a competent systematist, who gets to judge who's competent? This, as a practice, is still true today, in my humble opinion, when we talk about morphospecies. Not everybody agrees with this. Here's somebody who agrees with what I'm saying. Not to be authoritarian, but just to underscore that I'm not some weirdo. Well, I am a weirdo, as Jan Stevenson knows and Mark Edlin know, but this is not the evidence for that. But Whitmore said this very succinctly. The flaws Meyer attributes to morphological species concepts are not part of any modern species concept. So let's look at some modern species concepts. And again, for those of you who are deeply into this sort of thing, please understand that I'm not trying to, this is not a seminar on species concepts per se. I'm just trying to get to the core of each one, mainly to explain that morphology, there isn't such a thing as a morphological species or a molecular species. Let's start with what some people, and Dave Phillis here at UT says is the only concept, an evolutionary species concept. And I'll explain why he says that in a minute. Where a species is composed of organisms that maintain their identity from other such entities through time and over space. It has its own fate, it's bounded by speciation events. What that means is sort of like on a cladogram, something like that, a phylogenetic tree, that that's a species, that's a species, that's a species, that's a species. Whenever there's a speciation event, the original species dies and new species begin. There's more subtlety than that, but there's a Hegelian species concept in cladistics, and it's very much like the evolutionary species concept, sort of like a river through time, and it branches into various streams. We can look at some more specific ones. You can see that the evolutionary species concept doesn't say anything about data. The biological species concept, which is the one many of you are familiar with, it doesn't say anything about what kind of data are used. Its criterion is, of course, whether or not populations are interbreeding. There's a phenetic species concept. We're going to briefly mention, we're going to mostly focus on this one and another one. The phenetic species concept, very formally, that's the smallest cluster of organisms that a taxonomist cares to recognize. And when we talk about phenetic species concept with a capital P, we're talking about clusters that are defined or diagnosed by some formal clustering algorithm. Many of you ecologists are familiar with ordination procedures, clustering type procedures. A lot of these have their origin in the field of quote unquote numerical taxonomy, which attempted to classify organisms on the basis of overall morphology. Oops, I don't know why it's showing up like this. Of course, it's not restricted to morphological data. You can use molecular data with phenetic species. You can use morphological. You can use both. You can use physiological data. You can use behavioral data. There's no restriction. Biological species concept, as I've mentioned, you all know that. How it relates to these data, we determine whether or not a species, the concept rather, is about reproduction. Then reproduction, we can imagine, might have some consequences for the molecular or morphological or physiological or behavioral characteristics of our populations, of our species. And we might imagine that within an inbreeding population, the characters are more or less continuous. And then when things aren't interbreeding, that there might be some discontinuities in these observable features of organisms. But the biological species concept itself is not about data of any kind, heritable data of any kind. It's about reproductive isolation. So these are consequences of speciation. They're not part of the formal concept. And finally, there's phylogenetic species concept. I'm putting here Autapomorphic because, again, those who follow this literature will know there's sort of two flavors of the phylogenetic species concept. I'm focusing on the Autapomorphic here. And I'm going to do my best, by the way, not to judge between these and say one is better than the other, because I don't want to get into that. I don't want that to be a distraction from my larger point. The basic idea of a phylogenetic species is that species are no different than any other taxon. A species is simply, here's our tree, here's a monophyletic group, here's a monophyletic group, here's a monophyletic group. A species is just the smallest monophyletic group, the smallest taxon that we care to put a name on. And because in some way we draw our phylogenetic trees based on synapomorphy, the taxa are related in some way to synapomorphy, and an autapomorphy is a synapomorphy at the tip of the tree, that's all. So some way these taxa are related to a narrow definition of homology. I maybe should have put this early on. Don't confuse phonetics with a small p with phenetic with a capital P. Phenetic, and I'm not going to argue whether one's right or wrong, just that I want to point out that the word phenetic has been and often continues to be treated as a synonym to morphology. Phenotypic data, phenetic data, this means in a lot of places morphological data. It has another meaning, and this is the only meaning I'm going to use in this presentation. And that arose in the 60s. There's this formal arena known as phonetics, also called numerical taxonomy, which attempted to formally operationalize the ideas of similarity and dissimilarity using clustering and multivariate statistics. And again, it could use any kind of data. So when I use phonetics, I'm talking about this. So it's not restricted to morphology. So I'm not saying that's wrong. I'm just saying it has two meanings. Like a lot of words have more than one meaning. I'm using that meaning. Here's an overly simplified description for phonetics versus phylogenetics. That again, I hope for this audience, which I understand it to be often mainly ecologist or ecologist based. So when you deal with identifying species, you might use a key. A lot of keys will have a has a and a lacks a couplet. Valves heavily ribbed, valves without ribs. That's what I mean. In general, I mean having a to is in the rest of this talk is not just having a feature, but it has, we can say, the derived or the apomorphic feature. An example is the vertebrata. Vertebrates have a backbone. Vertebrates, as it turns out, are monophyletic, and that's one of the homologies or the synapomorphies for vertebrates. Lacking A is often the primitive condition. So invertebrata lacks backbones, and we know today that the invertebrata, any classifications that uses still invertebrata as a formal classification, is pointing to a non-monophyletic group. It's everything that's left over after you take out the vertebrates. Again, this is something of a cartoon, but I hope you get the point. By the way, I'm not seeing chats or anything come up. If anybody has questions, I guess direct them at Mark or David. And if y'all want to, go ahead and interrupt me, because I want to make sure people basically understand what's going on as we transition through this. So let's talk about how these things relate to lineages. We can look back at the evolutionary species concept, which is certainly a very generic concept. It has something in common with at least the biological species and here that should be the phylogenetic species. That these are sort of at two ends of the spectrum. The biological species says that species are the fundamental unit of evolution, whereas the phylogenetic species, and boy, that's not good, is it? Can't write on a screen because it's bouncing. The phylogenetic species at this end of the spectrum says that species are the smallest product of evolution. And this is very important that we care to put a name on. We have lots of branches in a phylogenetic tree. We don't name every single one. We name things according to human needs. But we're restricted to naming monophyletic groups that are diagnosed by synapomorphy, or groups that belong to a single branch. So species are no different than any other taxon. They require the same evidence. And they're the smallest product of evolution. So one end is about products, one end is about units of evolution, but both tie to lineages. Certainly when we talk about taxa, we're talking about lineages in some sense, these unique groups. And things interbreeding are keeping things together through evolutionary time, keeping these genetic links between individuals through evolutionary time. So they both relate to lineages in some way. I thought I had a slide here. I must have moved it or deleted it. The phenetic species, phonetics in general, is explicitly atheoretical. It's not theoretical. It doesn't tie to any of these things explicitly. It is just operationally defined. That's why I didn't mention phenetic species. There is no tie to lineages there. But we still have to be able to tell things apart And we can't go in our backyard and really see if birds are interbreeding or not. Almost all cases, we infer interbreeding from observation of characters. And so we ask how much separation of characters is enough, but we can even ask how much lack of hybridization, how much separation in terms of reproductive isolation is enough. Sometimes it's complete, but a lot of cases in nature, we know it's not complete. Mallard ducks form fertile hybrids with just about any other sympatric species in the genus Anis, and there are a lot: pintails, widgeons, gadwalls, teal. What keeps these things separate is behavior, not geographic isolation. But if the behaviors coincide, if they're in the same pond and they're reproductively ready all at the same time, you'll get mallards breeding with all of these things. And they all reproduce like mad in the barnyard. So bringing things into the lab and getting them to interbreed or not is not enough, because a lot of determinations of whether things interbreed and whatnot in the wild has to do with natural signals. So we're not going to talk about this much. We are going to focus, though, if we're going to be focusing on, my pen stopped working, there we go. If we're going to be focusing really on morphology and molecules, we need to focus on the phylogenetic species concept. And if you read a lot of literature, actual studies of speciation of things, a lot of it is certainly operationally involves the phylogenetic species concept. So it's not unfair to focus on that. We're going to intermittently discuss interpretations under the biological and phenetic species concepts as well. What are the values of varieties, forms, and other subspecies distinctions? Well, this has been debated a lot in the biological species literature, and basically there are conventions making it easy to sort of deal with this grade of this grade from no interbreeding whatsoever to some modest amount of interbreeding to a lot of interbreeding. Under phylogenetic species concept, they have no value whatsoever. I personally lean more to the phylogenetic species concept, but I think in both cases, they really don't have much value. Under the biological species concept, it's a coarse way maybe of dealing with this degree of interbreeding we think might be going on, but they really have no formal status in any of these concepts. They have a formal nomenclatural status, but they don't have a formal theoretical status. I hope that helps. So phonetics, here I moved the slide. Phonetics is explicitly atheoretical, so lineages are not involved at all. Phylogenetics and should say biological species concepts explicitly relate to lineages in some way. Okay, so we're going to go from words to diatoms now. This slide is not really about species and speciation per se. This is from a paper a student of mine did back in 2003, and I'm going to mention this to lead into using morphology in phylogenetic analysis. We don't see that done too much because, quote unquote, the characters are ecophenotypic or too variable or something. She did this study on what's called heritability. And probably just don't think of the word heritability, just H squared. Heritability is not what you think it is. I always have trouble with this in my evolution class. It's not how heritable the character is in an individual. It's talking about genetic diversity within and among populations. So the big H squared is 55 to 67% for total variation in a realer and stria density in a Lacocera subarctica. What this means is the populations are highly genetically diverse within and between populations. That is that these characters are reliably tracking underlying genetic variation. we can do studies like this on morphology to get an idea. This, by the way, in these kinds of studies is a really high number. So that's, we were surprised by that. So that's a good thing. What I want to get to here is the phylogeny of Alacocera as a whole that Stacy did. And this doesn't involve species and speciation. So I'm going to go through it very quickly. But I want to show you, it's one of the best studies I know of to show you how morphology and molecules and different aspects of morphology and molecules provide different levels of resolution. Okay, so that's all we're looking at right now. And what you're going to see, it's not really morphology versus molecules. So here's a phylogeny of Alacocera, a whole bunch of species using qualitative morphological data. Has spines, doesn't have spines, rows straight, rows spiraling, big areli, little areli, that sort of thing. Okay, and there's not a lot of resolution from the qualitative data. There are these nice little clusters, Sforzoii and niagarae, granulata plus muzinensis, a whole lot of ambiguous together, and so forth and so on. So some resolution, and this is what I mean when you have some parts that show some sort of branching and aren't just a big cone. All right, let's jump all the way to molecular data. We, of course, have fewer terminal taxa in this phylogenetic analysis because a lot of those Alacoceras are only known from the fossil record. Some of them we couldn't get cultures of, and so forth and so on. So taxon sampling in molecular trees is usually not nearly as dense as it could be with a morphological analysis. So that's one problem with molecular analysis. I do both, and there are problems with both. I'm not pitting them. In fact, that's a large part of what I'm trying to do here is to stop this pitting of morphology versus molecules. They're both data, and we need both kinds. So here's a phylogeny of Alacocera using SSU plus RBCL, and there are still large chunks of the tree that are not resolved. Here we can't tell niacensis from ambigua. Does that mean they're the same species? Not at all. It just means that SSU and RBCL can't tell these things apart. One of the things I want to point out is that in terms of barcoding, which is where a lot of the cryptic species talk has been using over the last several years, we've got several barcode segments in the SSU, and we've got the tail end of the RBCL in here. So we've got barcode genes in there. It's still not able to resolve some groups of species. Now here, one of the things Stacy did, and that's what the importance of this illustration, is Stacy used morphometric data that is measured as many different things as she could, the size of the hole, the distance the holes are apart. She had measures of straightness and curvedness and all sorts of things, depth of the sulcus and so forth and so on, the thickness of the wall, a lot of things like that that she measured. And so she was able to build a tree just with this morphometric data. You can see there's a tremendous amount of resolution. There are some odd things in it. Icelandica, Sforzoii are very separate from bicolensis. The qualitative data and the molecular data both, I think, group Icelandica and Sforzoii and put bicolensis a bit closer to those than we see in this data. So you can think of the morphometric data as perhaps resolving some of the tips a little better, but some of the deep branches, like here's the ambigua group, and muzinensis is separate. Things like that. We could go through the tree, but that's not really the point. Just that the morphometric data really have quite a lot of resolving capacity. Whether it's the right tree or not, it's a different question. So here's all the morphological data, qualitative and the morphometric data. And this big unresolved group goes away. We're able to resolve that by combining these data. We also see that Icelandica, Bicolensis, and Swartzii are put closer together. They're not joined together because we've been able to add a lot more taxa to this analysis. So molecular data make us think that these three species are very closely related. Well, if we consider extinctions and things we're not able to get in culture, there might be a whole lot of other stuff that's very closely related. Finally, here's all the data combined, the morphometric data, the qualitative, and both genes. And we see, again, Icelandica and Swartzii together, Bicolensis here, and all of these things. Basically, what this is a whole bunch of closely related stuff. These have big holes, these have little holes. But the real point, again, is the resolution increases as we look at different kinds of data and as we add these data together. The lowest resolving power was this one. That's the qualitative morphology. You can think of it getting back to species now. This is sort of the morphospecies approach. I've looked at these things a lot, then I'm an expert. I measured a couple of characters, arela density and stria density, but I didn't look at the morphometric relationships. Let's look at DNA sequence. Well, it gives us better resolution, so it must be better, right? Well, in fact, better resolution is obtained with morphometric data, and the very best resolution is obtained when we put all of these data together. And that's the take-home message I really want to leave you with, that there's scales of resolution depending on the data you're using, and different data might speak to different parts of relationships, and they might speak to different biological problems. So we're going to get to the last half of part three rather. We're going to address we're going to look at an actual what is a species problem. I'm going to focus on the niagarae complex, which I've been working with, as Mark said, pretty much my whole life. It was my dissertation, and I've returned to it from time to time as new ideas about how to analyze data have come about. So we have 4 species that I'm going to focus on. There's a couple more than that in this, but I'm going to focus on 4 species: niagarae, Yellowstonensis, superiorensis, and reimeri. These are all has-a species. They're all derived. They're diagnosed by apomorphy. All meet the criteria for phylogenetic species and phenetic species. Stephanodiscus niagarae, as I'll show you, these are kind of conclusions, lacks A. It's diagnosed by plesiomorphy, or primitive characters, and it only meets the criterion of a phenetic species. So let's look at take a morphospecies approach. Gene Stormer and I, the late Gene Stormer and I published a paper on this, holy cow, for more than 40 years ago. Geez, I'm old. Jan, are you that old? Probably.
No, I'm not.
You're not, okay. Jan and I have what's called white beard privilege. We can say all sorts of weird stuff because we're really old farts. He denies it, but he is. He was there.
Yeah, at least some of the time. Yeah.
Yeah. Well, he remembers. He was always there, but he only remembers part of the time. Of course, I only remember part of the time too, but that's a whole other story. So back in them days, using the morphospecies approach, again, vague, Gene and I couldn't really tell niagarae from Yellowstonensis, what today we call niagarae and Yellowstonensis. We felt they were different, but we felt conservative. Somebody else might have looked at them and right away said, oh, I think they're different enough to be 2 species, but that's just it. I think. Where are the data to demonstrate this? Well, a few years later, I did morphometric analysis of these things, measured, I think, 17 different features. Here's a diagram from paper and that I did that. By the way, I've got a bibliography at the end. Again, I'm still not citing each and every paper. This is just sort of a collection of mine and Gene's work. So again, here's niagarae, here's Yellowstonensis. What we showed is that a lot of the features that didn't look like they told these two species apart are strongly affected by diameter. And when you take that diameter effect into account, you can begin to see two different clusters. And when you, if you will, add up all these differences using multivariate statistics, you get two very distinct clusters. Here's Yellowstonensis. Here are all the things you called niagarae. The type is in here somewhere, some type or isotype material. So that's why that big cluster is niagarae. So by the time we finished those studies by 1987, it was clear that Yellowstonensis and niagarae were definitely good phenetic species, but it's not saying anything about whether there are lineages. Also, you might imagine that this smaller cluster is going to be harder to divide than that big cluster. So, you know, maybe we'll find cryptic species here, right? So good phenetic species in 1992, I did a phylogenetic analysis of the morphology of Stephanodiscus. What we showed was that niagarae lacks A. You can see that there's no characters mapped here. It's not my decision. After taking the characters that I identified, when you do the phylogenetic analysis, none of them map here. In other words, in terms of morphology, niagarae is no different than whatever would be at that node. You might call it an ancestor. So there's a hypothesis that it's an ancestor, whereas Reimeri, Yellowstonensis, and Superiorensis all have characters that are unique to them. So they all have A characters. Niagarae is a lax A. It is, remember, a phenetic species. Well, Yellowstonensis is as well, but having these characters says it's a good phylogenetic species as well, is both phylogenetic and phenetic. In terms of ancestor descendant stuff, we did fossil record in 2006. And sure enough, morphologically, about 14,000 years ago, when Yellowstone Lake first formed, when the glaciers went away, it became something that looks like Yellowstonensis and stayed something that looked like Yellowstonensis over the last 10,000 years. So niagarae is not phenetic, is only phenetic. It is not a lineage because part of it became some other lineage. It's not a unique lineage. Only Yellowstonensis and by extension Superiorensis and Reimeri are both phylogenetic species and good evolutionary species. All right, so yeah, just that. So which one do you think? We're going to look at the sequence data in a bit to look at something about phonetics and crypticism and so forth and so on and what's what. So we started looking at ITS data, internally transcribed spacer region in the ribosomal RNA cistron of from the nucleus. And guess what? Where we could distinguish Yellowstonensis, niagarae, Reimeri very well. I don't have any sequence for Superiorensis. With the eye, we could distinguish them morphologically easily. We can't distinguish them at all with the ITS. We had a lot of strains and we had a lot of clones of strains so that we could look at intragenomic polymorphism. They're all the same. In fact, you can get some strains, where is it? There's a pair here, I think, some pair, I don't know, right here. There's a pair here where one piece of ITS from niagarae is more like a piece of ITS from Reimeri. So not only do they not distinguish them, they can confuse these morphospecies. Don't worry about the others. Happy to talk to you about them after. But they show similar things. Look at another barcode gene, the COX-1. And again, right here, can't distinguish between niagarae and rhymerai. Let's look at the whole RBCL, which includes a barcode section, and once again, there's no difference. We'll look at the entire SSU sequence, which is going to have several barcode regions. It doesn't resolve, the SSU doesn't resolve a whole bunch of chunks of Cyclostephanos, doesn't resolve niagarae and Yellowstonensis. And if we look at just the SUV4 region, which is proposed barcode region, it doesn't even resolve Stephanodiscus and Cyclostephanos. It's incapable of resolution. This is not a consensus tree. This is the tree with characters mapped on it or character distances. There's no difference in the V4 barcode region between Stephanodiscus niagarae and Cyclostephanos dubius, Cyclostephanos WT16. So, oops, what do we have here? We have not just morphospecies that we can, morphological species that are well resolved, but we've got well, excuse me, my mouth is dry. Let me have a sip of coffee here. What we have is the opposite of cryptic species, of our normal thinking of cryptic species. These four barcode regions separately or together could not resolve species in the niagarae complex. Well, as Yellowstonensis is a good species, it had a 10,000 year record of evolving differently from niagarae. I don't know what else you can say, except it's a whole different thing, certainly a different lineage. But 4 barcodes couldn't distinguish it. Heck, barcodes, some of the barcodes couldn't even distinguish genera. So do we have moleculo crypticism? Why don't I just go ahead and come up with a whole new term and become famous for a new term? Woohoo, Ed gets to introduce a new term, moleculo-cryptic species. I beg you, Jan, Mark, please terminate me in some way. And I know, Mark, you have the capability to do that. Terminate me in some way if I do decide to publish this, because it means I've lost all touch with reality. I'm not interested in that. This goes back to the idea that different tools for different jobs, different tools have different levels of resolution, and you cannot predict when morphology good and molecules bad, or molecules good and morphology bad. Basically, molecular data generally resolve structure that's not revealed by these quasi-phenetic authoritative analyses of morphology. Okay, that's been beat to death, and I totally concede that, because these morphospecies are flawed at a much deeper level. Not that they're morphology based, but that they're not based on any sort of repeatable, analytical, operational, or theoretical basis. They are intellectually empty, and because they're intellectually empty, this notion of cryptic species must be evolutionary, intellectually empty. And I'm just going to close with a couple of more slides now. I think my point has been made, that even worse than being intellectually empty, this pursuit of trying to understand what a cryptic species is gets in the way of our understanding of the evolution and biology of diatoms. I will pick on this one. Many of you will recognize it anyway, but I'm picking on this one not because it's the only example, but because it's such a clear example. Finlay et al. 2002 denied that S. yellowstonensis was truly endemic. How do they do that? They dismissed morphospecies. And here they sort of did it in the opposite way. Well, these things are different, Yellowstonensis and niagarae, they accept that. But these two species are so identical that they must be really the same species, that this morphospecies, endemics is in quotes, the morphospecies concept failed. And in context, it's very clearly what they're saying. So by dismissing morphospecies, they dismiss, and by dismissing morphospecies in favor of DNA sequences, which we all know is better, right? This obscures the considerable support that morphological evidence, both cladistical and paleontological, gave to the hypothesis that Yellowstonensis is an independent lineage. So they, in my thinking, they have arbitrarily rejected Yellowstonensis as an example of endemism. And it's this approach to understanding cryptic species and weighing DNA over morphology that led to this, what I contend is an arbitrary rejection of endemism. So these things are really quite relevant to other questions, and focusing on crypticism hides this from us. And I'm going to close now with just a quick genomic view of niagarae et al. And this is data sort of hot off the press. We, Tricia Spanbauer, Joshua Cooper, and myself, have subjected 15 strains from 7 lakes and reservoirs in the Niagarae complex near Yellowstone Lake to next-gen sequencing, reconstructed the entire chloroplast genome, so forth and so on. I can talk to you more about that. Here's the real point, is that this is the tree we get. Very well supported. Lewis Lake strains, formaclade. Buffalo Bill strains, formaclade. Hebgen Lake strains, formaclade. Boison reservoir strains are kind of scattered all over, which is interesting. We don't know what to make of that yet. We only had one strain of Yellowstonensis, unfortunately, one strain from Jackson Lake. But here's one take-home message. All of this is niagarae. So at least for these data, this phenetic species is clearly not monophyletic. Another thing Gene Stormer told me, and he may have said it to Jan and others, when we start looking in detail with molecular data and morphometric data, every lake is going to have its own species. Well, Gene, you might be right to an extent. Some of these look like they are blowing all over. Idea that things, diatoms disperse readily. We still don't know what's going on with boison. We've got mitochondrial data and we've got nuclear data coming online. From the diploid area of the genome. So hopefully we'll be able to sort some of that out. So once again, this phenetic niagarae has no biological reality. The commonality of morphology doesn't seem to be due to interbreeding. Let's go back and look at Lewis Lake. I mean, heck, the strains in Lewis Lake are distinct from one another, and they're very distinct from lakes that are right next door. In fact, Lewis Lake flows into Jackson Lake. And there's a whole bunch of characters that separate Lewis from Jackson. So yeah, it doesn't look like these things are interbreeding. Maybe there's some going on with boison, but Buffalo Bill strains are virtually identical. Hebgen Lake strains are virtually identical. So these individual strains, if we had to go with just these data, and there's real problems with just using 3 strains and but if we had to interpret those data, we'd think that maybe Hebgen and Lewis Lake really are their own species from a biological standpoint. But here's my real point. Some data resolve some problems better than others in some situations. You absolutely must use formal analysis, especially with morphological data, to observe that. Dismissing morphospecies and morphology, you do that at your own peril. So I think I've made my point. Let me just close with this just to explain something. I'm just showing this enough so it's on the YouTube video because I want to show bibliographies. You'll have a chance on the YouTube video to write these down. I won't say anything about it, except what I'll say is that this is very much a partial list. Honestly, I kind of slapped it together. I had a major migraine headache for about a week and a half that concluded itself Sunday. I don't claim this to be authoritative in any way, not even for the niagarae work for any of these topics, but somewhere in all of these papers, if you want to know more about it, and if you want to know details and want to get to primary literature, all of these papers somewhere along the line point to primary literature. So if you're interested in this stuff, this is a good place to start. And here's my contact information, and I am done.
All right, thank you so much, Ed, for gosh, making us all think about these concepts and the limitations they have and where they're going. Actually, if people would like to ask questions, we got our group isn't we got a little over 30 people on board. If you have questions, why don't you type something in the chat? We can bring it up there. We'll call on you and have you on mic. If you do have a question, you don't have a mic, just write your question and just write no mic or something like that, so we don't have to fight through that battle. As people start asking some questions here, I want to just ask one question of you, Ed. Number one, I thought it was interesting. One of the things you said is that we name things according to human needs, and it reminded me of something that I don't know if Gene said or Charlie said, but I think it may have been Charlie that it said something along the lines of the diatoms have no trouble figuring out who they are. It's we who have trouble figuring out who they are.
Yeah.
And anyway, let's we've got Jan's got a question. Go ahead and unmute yourself, Jan, and go ahead and ask Ed questions.
Hey, Ed, thanks a lot for the great seminar. Can you hear me? I've got my headphones in.
I can hear you.
All right. So thanks. I really enjoyed it. It's the classic great scientist kind of thing where you're always thinking and asking questions and assume you don't know anything.
I can pretty much prove
wide
that
open.
I don't know much.
Yeah, well, the idea of knowing versus I want to figure more out. I guess the idea of using ecological data as well as molecular and morphological data and describing and defining lineages. I've got a question about that. And I think that's what ecologists do a lot, because it's not really practical for us to do molecular analyses for all the morphological entities that we stick names on and then look for patterns with environment. And those patterns in the environment are related to their physiology and anatomy, even microanatomy. And that, of course, is related to genes.
Yeah.
So, I mean, you just kind of got into some of that where different lakes have all these smaller lineages.
I can come back to actually give a seminar on that at some point if y'all would like, but I'll say this as a conclusion. If systematists, taxonomists would do their job right, and in the morphology, which ecologists have to deal with, as you say, most of the time you got to deal with the morphology. If taxonomists would do their job right, they would tell you, okay, Yellowstonensis, and they would tell you Yellowstonensis and niagarae are both good phenetic species, but only Yellowstonensis has apomorphies. Okay, we would tell you that, and then the ecologists would know Yellowstonensis, the thing with the apomorphy, is a natural unit, and so its ecology is probably narrower and identifiable. But the plesiomorphic thing, I need to take that range of pHs and that range of salinities and so forth and so on with a grain of salt that it's not going to be as reliable or exact. The niagarae example is an extreme one of that. I can tell you quickly, you look at Benderanus and Hansii. Benderanus is got a lot of derived morphology relative to Hansii. They are in those clusters, indistinguishable by the barcodes, but Benderanus certainly has a derived apomorphy, and it's not like Yellowstonensis, just in one place. And when you plot those two in ecological space, the ecological space is defined by Hansii, and Benderanus occupies a little narrow corner of that.
Yeah.
So the apomorphic species is still your best signal for ecology. So if you're looking at a core, you might decide, I'm going to build my my transfer function not using all the species, just using the apomorphic species. And that's not the ecologist's fault. That's the fault of us systematists for ignoring the work of Hennig for the last 60 years by not incorporating that into our thinking.
So
We should.
It seems like, though, you are saying that the ecological data is actually helping confirm, and in some ways,
probably. I know other people have questions, Jan. We can. I don't mean to cut you short, but it's more. Long and short of it is, figure out your natural units, then look at the ecology. Where the ecology is important, I'll just say this, is to support the hypothesis that Benderanus arose through peripheral isolation. So it's not without value, but for the strict ecologist, if we did our job right, you guys might decide just to work. You would either decide to work with just the apomorphic species, or you would take into account which are apomorphic and which are these plesiomorphic, just phenetic species, realizing they may not be natural groups after all. But that's on us, not you guys.
Thanks, Jan, for that question. Ed, one of the questions I had sort of looks at this contrast between Aulicosyra, Stacy's work on Aulicosyra, and the continuing work on Stephanodiscus. We saw we saw difficulties with morphological data, or we saw differences in morphological versus molecular data, but great resolution when we combine those two. Have you been able to try that same technique with Stephanodiscus, where you sort of had the opposite results with Stephanodiscus?
Well, we're sort of doing that now by focusing just on niagarae. I don't have a complete, and over the years, as I've gotten nifty looking Stephanodiscus, and as Andy Alverson has gotten nifty looking Stephanodiscus, we've been sequencing them. So before I retire in the next 4 or 5 years, we're gonna do that. But right now it's been mostly about niagarae per se. But you know, we already see the same thing there, that some of these barcode genes are just going to crush stuff into nothingness. But they're the morphology. I mean, like Cyclostephanos. Cyclostephanos has a couple of apomorphies. Stephanodiscus has a couple of apomorphies. And the V4 region can't tell them apart at all. Can't tell genera apart. If you just threw out primers in water for the V4 region, you wouldn't know if you had Stephanodiscus or Cyclostephanos, and I don't know, maybe Discostella. I don't know.
Sure.
I haven't added that.
Nick Schulte posed a question in the chat. Says, thinking of single amplicon DNA sequencing or metabarcoding, what are the dangers of assigning species names that likely have been derived according to different concepts, or going taxonomy free and just looking at sequence similarity alone when defining taxa for community-based analyses?
All I can give you there is a quasi-informed opinion just Shona Manning is doing a lot, and we talk a lot about this. Number one, we need much better voucher databases, and Shona is starting to work at that. She just got a job at Florida International University. Hi, Tom. And that's one of her goals there, is to do just that. I think both are useful, because there's going to be a lot of stuff we get out of environmental metagenomics that's just not going to go to anything. There's going to be things that mash genera together. I think there is value in dealing with ribotypes, but if you can reliably identify some species, then you get to take advantage of the work that Jan and you and many others here have done. That's not just ribotype XYZ number 325, but that's species such and such, which we've done a lot of ecological work on. Mm-hmm. That's all I've got, really.
Okay. Well, thank you. And I want to again extend my thanks. Whoops, we got one more question. We've gone a little bit over the hour, but I think we're okay. Everyone's okay with that. Tom, you want to unmute and ask the question? I can see you.
Hi. Hi, Ed.
Tom and I have been emailing for about a jillion years, and this is the first time we've seen each other in at least 2 dimensions.
Oh, wow.
Yeah, for sure. Yes, I was asking, do you argue for a formal qualitative analysis with every new species description?
If I was in charge, I would require it, yes.
Okay.
But what I do in reviews that almost always get ignored is insist at least on careful and formal phonetic work. Don't just report stria density. Don't just report diameter. In fact, stria density is, I could go into that separately, is a terrible character. Just report the total number of striae. And like Mark Edlin did, thank you, Mark, the only person who's ever paid attention to me in this plotted number of striae, number of virili, and so forth and so on against diameter. So at least do good morphometric work when you report those numbers, because those things are always overlapping, because those things are always related to size in some way. But yeah, if I could, I would. Although for a lot of groups, it would be difficult probably, but I think it would be wise. I think people ought to do it as much as is feasible.
Sure.
You're going to be wrong sometimes, but all of these things, everybody always says, well, classification's just a hypothesis. Well, yeah, let's just treat it as that and be as formal about generating our hypotheses as we can be. Don't just say it's too hard, I'm not going to do it.
Sure.
That's me.
Well, thank you, Ed. Really appreciate you coming on the Diatom Web Academy and providing us with a really interesting talk. I do want to remind everyone that in 2 weeks, Elizabeth Alexson is going to be speaking on Great Lakes Fragilaria. Please join us then. We'll also have Ed's talk up, we've been recording it. It'll be up on our YouTube probably within the week. Again, you can find links to that at diatoms.org and go to the news page and track down links to these things. Thanks again, everyone, for joining us, and have a good rest of your week.
Thank y'all for having me. And good to see you guys.
Yeah, and I just want to say thanks again for a great talk. It was very stimulating, and Nick Schulte had also had conveyed, and a lot of people in the chat are saying thank you. But yeah, great talk. Appreciate it.
It was fun. Thank y'all. And it's forcing me, I've got about half of that is unpublished, and it's forcing me to get off my ass and do that. All the ITS work and stuff, I actually had submitted it, and then I had a ascending aortic aneurysm.
Oh my gosh.
And had that repaired, and that kind of slowed me down for about 3 or 4 years. Just at the time the museum closed and I was thrown into teaching full time. So I went from being an evil administrator to teaching giant PowerPoint classes And of course, I knew the material, but as any of you who teach know, taking what you know and putting it into a formal teaching package is a hell of a lot of work. And also you find out how much you don't know
Sure.
when you start to do that.
It's a great way to synthesize and think, isn't it?
Yeah, yeah. It's really had a lot to do with my thinking. Talking about ecology, it's really made me think about peripheral isolation. And so I started looking for other examples. And that Hansi benduranus one is great because they're so closely related. A lot of people think, well, maybe they're the same species, so there's no argument there. And I'm plan on getting benduranus. I think you can't misidentify it. A lot of the Hansi might be, but it's still gonna look like that, very widespread. I'm gonna contact the people where the NLA slides are kept and look at about 10% of them to get an idea of how much error there is in counting Hansi. Yeah, I mean, this idea that the peripheral isolate is going to have evolved in an ecologically edgy area like the niagarae stuff in my systematic biology paper fits benduranus and Hansi perfectly. But it's more interesting because since that peripheral isolation, unlike Yellowstonensis, which hasn't dispersed yet, apparently, benduranus certainly has. So, yeah, you're looking at there an event that might have occurred 100,000 years ago, but you're still able to detect the signal by, I would say, forgetting about morphose species and just understanding, looking at plesiomorphy, primitive versus derived, yeah, just doing that. So again, but that's on a systematist. I hope I made it clear. I don't blame the ecologist for this or the ecological perspective. In a nutshell, y'all got to work with what the systematists do, but people who call themselves systematists in diatomology, and David Williams is on here. He and I would debate a lot of these issues, I'm sure. And I hope, David, you were okay with my sort of general fuzzy presentation. As I said,
the ecologists do blame you guys, though.
Oh yeah, absolutely.
You know, you don't blame us. We do blame you.
And you should. We have not done our job. Willie Hennig came up with this stuff and it was published in English. I don't know. He came up with it, I guess, in the 50s, and it was published in English in the 60s, and it's all over systematic biology and systematic botany and everything else. And diatomists have David and I would, I think 100% agree on this, that diatom systematists have done their absolute best to ignore it. Yeah, it's our fault. It's not your fault.
So we've got a lot of good things to do in retirement.
Yeah. Well, but it's even worse now.
I want to encourage you when you take this next look at Stephanodiscus. You know, I found it very interesting that the Parvis and Minuculus were so far apart on the trees, and you know, I'm not terribly surprised. But you know, those are two. There's several texts that I see often co-occurring. I see this in Salafra, and I love that you're like, don't pay attention to striate density, because I've had some suspicions about these Levissima. There's like a dense form and a coarse form, but they're everything else the same about them, and what I see ecologically and the large number of lakes and cores that I've looked at so far. But you know, within Stephanodiscus, I always see Parvis and Minuculus co-occurring. You know, I've tried very hard to be very careful and try to tease out ecological differences, and I just don't see them. So if you could, for me, pay attention to that group a little bit when you're doing your next analyses, I'd appreciate it.
Yeah, there's a famous paper on that.
Two things I can say off the top of my head that I remember, I think, accurately. First of all, this I know for sure. I have looked at the type material of Parvis. Unfortunately, it's just a slide. There's no material to do SEM of. It definitely has a central area strutted process. Hannelor's, or whoever it was, distinction that it was isotopic in one and heterotopic in the other. Nope. Both in the Parvis type material and everything I've undergraduate in my lab and I published a paper on this some time ago using morphometrics. No, there's there's no difference. There is when I look at it, it looks different. And the parvus that I've identified for the DNA is my best attempt to identify it based on observations on that type material. I can tell you one little diatom, one little Stephanodiscus that is easy to pull out when you're looking at it. When you look at these little Stephanodiscuses, take a little time to look at them and look to see if you've got one costa, one rib, that's noticeably thicker than the others. If you do, I'm 99% sure you're looking at what we call vestibulus, which is actually triporous. And I've seen a lot of samples and you can see that pretty good. If you get a flat specimen that's unobscured, that one costa that's thicker than the others, and it'll have a spine on it that's thicker than the others, that's the labiate process. So if you can see that, send me some pictures and I'll help you verify. But you can tell vestibulus. Then I've looked at material after that, looked at material in the SEM, and I'm always been able to get that. So that's one tiny step that you can pretty reliably distinguish.
I appreciate that. Yeah, it's one that I feel like has been maybe lost a little bit in the literature because I just don't see it frequently reported.
I think a lot of people haven't picked up on that, but I've had people send me small steps for identification, and I tell them, well, it's not just one, you have at least two. So it's more common than it's reported. I've seen it in bayous in Louisiana, reservoirs in Tennessee. It's been reported from Japan. I think they call it prey Stephanodiscus. I hate prey names. Yeah, that I always joke that prey it's Stephanodiscus. It implies that it's ancestral. And no, if it's here from a systematics point of view, no, if it's got synapomorphies, it can't be ancestral to anything. It's got to be like niagarae. It's got to completely lack characters. It's got to look like the primitive form, which means it can't have apomorphy by definition. It can't have its own homologies. If it has its own homologies, then it's its own branch. Again, but that's our fault, the systematists. That's an example of what I'm talking about, that people have done their utmost to not read this stuff. That's like cladistics 101. If I taught a cladistics course and I taught you what synapomorphy was, the very next thing I would say was, no, you can't do that. Not if they have even if they don't have apomorphy, that doesn't mean they're ancestral. It means they could be, but if they have an apomorphy, then they can't be. Or either they can't be or your cladogram's wrong. That's always, you always have those two hypotheses.
Well, thanks. I really appreciate it. And,
Sure.
and also let's talk and figure out when we can get you penciled in to come back.
Yeah, the whole peripheral isolation thing, that Bindorannus hansii story is, to me, really fascinating. And where it comes out ecologically, by the way, it's kind of weird. It's in smaller, relatively more oligotrophic areas. And that's one reason it's got a narrow distribution, is oligotrophic lakes tend to be big. There's not a lot of smaller eutrophic lakes and rivers.
So the other thing is that, I mean, from an ecological perspective, and from that oligotrophic systems are much more stressful for the organisms. They're really low nutrients. And being adapted to live in those low nutrients requires sacrifice for a lot of other attributes, presumably if you have trade-offs. So along those lines.
Well, that's why I say don't to test that kind of thing, Jan. This is why I say do the evolution first. Because if you build the ecological stuff into your classification, then you can't turn around and test it.
Right, but I can't turn around and test it with molecular.
Yeah.
If I'm doing morpho and eco, then I can test it with molecular.
I say I think you're better off doing it with morpho and moleculo.
You can't do molecular at the scale that we're doing it, where you're tying it to specific entities. I mean, you can when you find something really interesting and then dive into it.
Well, no,
and I can't do it. I'm leaving.
Yeah. Well, what I'm saying is you don't even need
a microscope.
Well, the Hansen-E thing, in fact, requires morphology.
Yeah.
To test that, because maybe once we get the whole genome, but for all the barcode stuff and 6 or 8 single genes, you don't get any resolution. Those things evolved in the last 100,000 years. And most of the sites that you're going to look at that are different, when you're looking at RBCL, okay, that has nothing to do with, almost certainly has nothing to do with niagarae evolving from, or Yellowstonensis evolving from niagarae, okay? And so selection is not going to happen. So the only thing that's going to work is genetic drift, neutral mutations, neutral substitutions, and they don't happen at that rate. You're not going to find many neutral features, many neutral molecular features that can tell you about things that happened 10,000 years ago. In the chloroplast genome, we're finding some, but that's because we're looking at 180,000 base pairs. And so among those, we're finding a couple of dozen substitutions. And by the way, the tree I showed you actually includes hotspots, like a whole 5 or 6, 8, 10 spacers. It's including those as individual indels, individual changes. Well, arguably an indel like that is just one event. And so when I have an analysis, when you reduce that, you actually reduce the resolution a little bit. So even with 170,000 bases, we're just getting barely enough to get a tree. We got the mitochondria, which is about 80,000 bases. And it pretty much has the same tree. It has one interesting difference. So it flips, I can't remember what, it flips a couple of things around, which suggests that they're differently inherited. Oh, Mark, we cite your paper with Gene, and y'all where you saw the chloroplast in both, but couldn't say anything wisely, I think based on your observations, about whether it was uniparentally inherited or biparentally inherited. There are tests to see if, so the chloroplast genome is a single locus, and there are tests to see if there's been recombination. And we got some weak evidence for recombination by these statistical tests if you include every point of these hotspots. But when you reduce them to a single evolutionary event, it all goes away. So it looks like uniparentally inherited at this point, or let's say more accurately, we don't have any strong evidence for biparental inheritance. But that could explain something of the poison being scattered all over. We'll know more about it when I said there's a danger in just looking at 3 strains. We need to get more strains, and we need to look at them in different years. And that's going to tell us a bit more about the inheritance, whether it's uniparental or biparental.
Ed, did you find, it looked from your tree that you found niagarae in Lake Yellowstone or Yellowstone Lake?
No, it's just Yellowstonensis.
So that's nested in there.
Yeah,
okay.
It's nested in there.
All right.
Here's a thing though. Tricia went to Yellowstone about 4 years ago, Didn't find niagarae in June, July, when it's usually at its peak. Tricia and I went the year after that and didn't find any.
Ooh.
And I mean, we looked hard. I pulled plankton net toes at various places, and I looked at every drop of water in those plankton toes, and it's a big diatom. If it was there, I'd have seen it.
Mhm.
Didn't see it. My son and I went the year after, same thing. Didn't find a one. We did find one live cell. Tricia and I found one live cell, and it was niagarae. Unfortunately, and I isolated it, it didn't grow. But it was clearly the niagarae morphology. It was a big diatom, had one spine every other costa, which is kind of rare. Some people, I know a couple of people are trying to tell me there's Yellowstonensis at this lake and that lake. They send me the material. Nope. Because you just can't do the spine to costa ratio.
Mhm.
The spine number declines at a different rate than costa number. That means the ratio between spines and costae is itself diameter dependent. This is why these ratios of stria density don't work, because those densities are diameter dependent. So it turns out the raw numbers are perfectly linear related, but
Mhm.
these ratios are always some weird curvilinear relationship unless the intercept is zero, meaning if it was one micrometer in diameter, it would have one spine. If it was 2 micrometers in diameter, it would have 2 spines. No, they're always like 8 to 15 or something like that, which means a diatom of 0 micrometers diameter has 15 spines. So if you see 15 spines floating around, that's the cell. But that relationship lends itself then, when you make a ratio of those things, it always makes this weird curvilinear relationship. So why not analyze the easy to analyze linear relationships? There's also a measurement problem, Mark, as you know. Any of you who've tried to, if you measure a big centric diatom, then your tangent is to the circle. You know, your circle's like this, and your tangent that you're measuring on is like that. No, they're the same thing. But how do you measure the number of striae in 10 micrometers with a 9 micrometer wide diatom? You know, but you can count the number of costae, the number of striae. If you think about a bicycle wheel, you know, bicycle wheel, if it has 100 spokes, it's so many spokes per 10 micrometers if it's this big, but it still has 100 spokes, and you make it that big, and now you got a lot more spokes per micrometer.
So Ed, are you in favor of circumferential density?
Who what?
Circumferential density, where you count all the
Nope.
No?
You still have the same problem.
Okay.
Yep, you still have the same problem. You've got the measurement error, and you've got this problem that the bicycle is trying to illustrate. That you reduce the circumference, but you still have, if you still had 100 spokes,
sure,
now you lose some costae, but if you don't lose enough of them, if your intercept is not 0, you're still going to have spokes, a lot of spokes, when you're near 0 micrometers in diameter, and diameter and circumference are the same thing.
Yeah. All right.
Yeah. So yeah, numbers per 10 micrometers suck. Point blank. Don't do it.
Don't do
it. You can do it when you get out to really big stuff. The slope is more like this. But if I published a paper, Dave, email me and say rotational symmetry.
Okay.
And what you're going to see is, and this is from empirical data, I measured all kinds of things, labiate processes and this and that, and this and that. And everything that on all kinds of centric diatoms, and everything showed that relationship, linear with size reduction and curvilinear with ratios. And that all the intercepts were all like about 6 to 15. What this translates out to in that curve is that anything below 20 micrometers, the density is going to change wildly with just a couple of micrometers difference in size. When you get out to 30 or 40 and above, it barely changes. Certainly your measurement and the diatom, stochastic development, and so forth and so on. Yeah, it's numbers per 10 micrometers is going to be pretty good. That's also where your measurement error goes away. But when you get down below 30, especially below 20, then everything kicks in like crazy. Your measurement error and this nonlinear relationship go nuts. So yeah, above 50, go ahead. Between 30 and 50, be careful. And below 30, get ready for weird shit to happen. That's the rule of thumb. Yeah.
I'm gonna need a big thumb to write all this on, but
Oh yeah. 50.
All right. Well,
50, okay.
I gotta take off.
Real simple. Above 50, good. Below 20, bad. Between 20 and 50,
Anyway,
Thanks a
I
lot,
want
man.
to say thank you, and I gotta take off.